Yeah, good morning. We are going to talk about respiration. In the last lecture, we were talking about photosynthesis. Now, in respiration, glucose, which is produced in photosynthesis, is now utilized to produce energy. Basically, we have carbohydrates plus oxygen on one hand, producing carbon dioxide, water, and energy. So I'm going to make a presentation on respiration. So we'll talk about respiration. Now, the objectives on the lecture on respiration. We want to understand how ATP can be produced from glucose. What is glycolysis? What are the products of glycolysis? Pyruvate, acetyl coenzyme A are some of the products which we will talk about. We should understand that respiration occurs in the mitochondria. What are the key products of citric acid or Kreb cycle? ATP, NADPH, FADH2, and CO2. We want to understand the electron transport process, which produces about 32 ATP. We wanted to understand the key processes in respiration. Number one, glycolysis. 2, citric acid cycle or Krebs cycle. Electron transport chain. So what is respiration? Living cells require energy to drive biological processes. Energy has to be released from trapped chemical bonds. For example, glucose has to be broken down in order to generate ATP, which is the energy required for growth and other functions within the cell. We have NADH and FADH, which are used to generate more ATP later. So basically, we have seen this equation where we are saying carbohydrates plus oxygen gives us carbon dioxide, water, and energy. You could have come across this equation. Respiration is basically oxidation and reduction processes. Energy is transferred from food molecules to energy carrier molecules in the cell. Electrons are removed from donor molecules and passed on to acceptor molecules. Mitochondria is the organelle involved with the cellular respiration. With photosynthesis, it was the chloroplast which was important. Now with the cellular respiration, the mitochondria is the organelle, which is important. Yeah, let us look at the relationship of photosynthesis and respiration. Photosynthesis makes glucose that is used in cellular respiration to make ATP. The glucose is then changed to carbon dioxide, which is also used in photosynthesis. The two systems depend on each other. Here we've got photosynthesis in green and respiration in brown. So you can see the chloroplast, which is the organelle used for, the organelle which is used to produce glucose, and the mitochondria, the organelle which generates power in the form of ATP. Here we've got photosynthesis, which produces starch. Then we have got glycolysis, which produces the pyruvate. And we have got the Kreb cycle, which produces NADH, FADH2, and also some ATP. This is now respiration, which consumes oxygen and releases carbon dioxide. On the other hand, photosynthesis consumes carbon dioxide and releases oxygen. So on one hand, we've got photosynthesis, we have got the cellular respiration, we have got light-dependent reactions in photosynthesis, which emit oxygen. Then we have got light-independent reactions leading to glucose production. On the other hand, we have got breakdown of glucose via glycolysis, producing products which are required for Kreb cycle. These will utilize oxygen from photosynthesis. And then we have also electron transport chain, which produces energy. The Kreb cycle will emit carbon dioxide, which may be utilized by photosynthesis. So the breakdown of glucose occurs in 3 stages. Number 1, glycolysis in the cytoplasm, Kreb cycle in the mitochondria, then the electron transport system, which is located in the mitochondria. So these are the 3 areas we are going to focus on. Now, if we begin with glucose, it undergoes glycolysis using 10 successive reactions to form pyruvate. Now, depending on the conditions in the living system, if we have anaerobic conditions, for example, in animals or plants, lactate can be produced, or lactic acid fermentation. That is one route. You might have had some cramp after an exercise. It could be due to lactic acid accumulation. This is due to anaerobic respiration. Then we also have anaerobic respiration, which can be used in industry to produce ethanol and carbon dioxide, alcoholic fermentation. This one is possible through the presence of yeast, but otherwise, the pyruvate may be oxidized during aerobic respiration in the presence of oxygen and produce acetyl coenzyme A. The acetyl coenzyme A will now enter the citric acid cycle, which also utilizes the oxygen and later produce carbon dioxide and water. So if we start with glucose, we have glycolysis and the production of pyruvate. Then pyruvate oxidation to produce, this is the step which requires oxygen to produce acetyl coenzyme A. Acetyl coenzyme A will initiate the citric acid cycle by combining with what is known as oxaloacetate. Then what is produced is FADH2 and NADPH, which are later used to drive this electron transport chain. So glycolysis is the first stage, where glucose will produce two molecules of pyruvate at the end of glycolysis. Pyruvate could follow anaerobic or aerobic pathway, like what I've just explained. Anaerobic respiration, this can lead to ethyl alcohol and carbon dioxide. Aerobic respiration can produce pyruvate, which moves into the mitochondria, where it is oxidized into carbon dioxide and water. Then energy is trapped in ATP and NADH. There are actually 10 steps to convert glucose to pyruvate. ATP is consumed in steps 1 to 5. ATP and H are produced in steps 6 to 10. Here we can, I hope you see the case. We're starting with the glycolysis. We're starting with the glucose, which has 6 carbon atoms. This is phosphorylated using ATP. We get glucose 6-phosphate. Then it changes to fructose 6-phosphate. Then we have phosphorylation again using ATP to ADP. That is fructose 6-phosphate undergoes phosphorylation. Then we are now having fructose 1,6-phosphate. This will then produce 2 carbonate molecules, one glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. When we were talking about the Calvin cycle, glyceraldehyde 3-phosphate was the product which was used to regenerate ribulose 1,5 bisphosphate, and also 1 molecule of glyceraldehyde 3-phosphate was used to generate sugar, to produce sugar. So glyceraldehyde 3-phosphate, it's like we are working in reverse in the dark reaction. So glyceraldehyde 3-phosphate is reduced. Actually, it gives NADH. It produces NADH when it changes from glyceraldehyde 3-phosphate to 1,3 bisphosphoglycerate, which has got phosphorus at both ends. Then this 1,3 bisphosphoglycerate is phosphorylated. No, it actually produces ATP when it changes to 3-phosphoglycerate. So things are working in reverse at this point. Then we produce 2-phosphoglycerate from 3-phosphoglycerate. Then 2-phosphoglycerate will give us PEP. Then from PEP, we get pyruvate. And we have pyruvate kinase, which changes this step. And I must say there are a lot of enzymes which catalyze these reactions, which I've actually deliberately omitted so that we focus on these products. So this is how pyruvate is produced, starting with the glyceraldehyde 3-phosphate. Then we go to 1,3 bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, PEP, then pyruvate. And please note, at the first stage, we are making 2 molecules of NADH. And immediately after, we are also making 2 ATPs, and the last reaction also gives us 2 ATPs. So we could be producing 4 ATP plus 2 NADH using this last 5 steps of glycolysis. Well, this cycle was discovered by Hans Krebs, who was working at the Sheffield University. This is the current building at Sheffield University in the United Kingdom. Towards the end of this building, you have this other place. This is where there is the laboratory where Professor Hans Krebs was working. And he came up with this cycle. Krebs cycle, or tricarboxylic acid cycle, TCA cycle. Some people call it the citric acid cycle. Produces ATP, NADH, and FADH. NADH and FADH are further used to produce more ATP in the electron transport chain. So here we've got the pyruvate from glycolysis, which gives us acetyl coenzyme A, and coenzyme A may actually produce carbon dioxide here. Here we are also producing NADH. So acetyl coenzyme A will combine with oxaloacetate to start the Krebs cycle. The product which is produced is citrate or citric acid. It is citrate. Then next we get isocitrate. And at this stage again, NADH is formed between isocitrate and the alpha ketoglutamate. Between alpha ketoglutamate and succinyl coenzyme A, we produce again NADH, and the carbon dioxide is released from coenzyme A. Then from succinyl coenzyme A, we get succinate. Coenzyme A is released here. Here we have also GDP being converted to GDP and again to GDP. There may be also production of ATP. Then from succinate to fumarate, at this stage, another product, FADH, is produced from fat. Water is added, then we have a malate. Malate yields oxaloacetate, and then the cycle will start again. Between malate and oxaloacetate, we're also producing NADH. Well, if you want to remember all the products in the citric acid cycle, you may use this key. I've colored the first capital letter. Our city is kept safe and secure from mobsters. O representing oxaloacetate, C citrate, I isocitrate, K ketoglumarate, alpha ketoglumarate. Then succinyl coenzyme A, we've got S. Another S representing succinate, F fumarate, and M malate. Maybe this may help us to remember this list of chemicals. But otherwise, we are just interested to know the products of the citric acid cycle and also the products which actually initiate this cycle. Then we move on to the electron transport and ATP synthesis at inner mitochondria membrane. Remember, ATP is the energy carrier used by cells to drive living processes. NADH and FADH must be converted to ATP. Oxidative phosphorylation takes place here. We have the flow of electrons from NADH and FADH2 to oxygen. So there should be a 2 here. So we have the electron transport chain, which has got 4 multiple complexes. Complex 1, complex 2, 3, and 4. Here I've got a diagram showing the electron transport chain. In this case, we need NADH and FADH2 So I will explain what is happening here. We have got complexes 1 here, complex 2 here, complex 3 here, and complex 4. These are the parts involved with electron transport chain. And on the extreme end, we've got ATP synthase, which is involved with the ATP production. So what happens is that, this is actually taking place within the mitochondria. So what is going to happen is that NADH from the Kreb cycle will lose an electron and passes it to the complex 1, and it is converted now to NAD. Once the electron moves across the complex 1, hydrogen will be pumped to the top. As the electron crosses this complex, hydrogen ions are pumped upwards. Let's go to FADH2. It will also lose an electron to complex 2. And that electron is taken by Q or quinone, and it is converted to FAD. Quinone is a protein from NADH and from FADH2 to 3. Once the electrons cross complex 3, hydrogen ions are pumped upwards to the top here. Now the electrons are now taken by cytochrome complex and passed on to complex 4. As they reach complex 4, hydrogen ions are also pumped upwards. Finally, the electrons are accepted by oxygen, which will split and then combine with the hydrogen ions to form water. So at the top, there will be an accumulation of hydrogen ions, which will later pass through the ATP synthase passage due to differences in gradients. So as the hydrogen ions pass through the ATP synthase, it's like a turbine generating electricity. They tend to generate a lot of ATP. So that is what is happening in the mitochondria, where a lot of ATP is now being generated. So like what we said in complex 1, there's transfer of electrons from NADH to coenzyme Q or ubiquinone. 4 protons are pumped to the matrix. These are the hydrogen ions. These are pumped to the inner membrane space for every electron passing through the complex. In complex 2, FADH2, let's put a 2 here. And save. FADH2 passes save again. Passes its electrons through this system. But this system does not pump electrons. That is complex 2. In complex 3, cytochrome C complex oxidizes reduced ubiquinone. 4 protons are pumped to the complex by this complex, so the hydrogen ions will be moved to the top. Cytochrome C then transfers electrons to complex 4. Complex 4 now moves electrons, which will reduce oxygen to 2 molecules of water. So oxygen will accept the electrons, and then water is formed. Now, the role of ATP synthase. The hydrogen ions pass through the ATP synthase and cause the conversion of ADP to ATP. This system produces, let's see here, 32 molecules of ATP. So a lot of molecules of ATP are generated by this system.
Matrix of the mitochondria of all our cells is the cycle of reactions that one hand Krebs a Nobel Prize. The cycle takes the energy in food and makes it into other forms your cells can use. Krebs cycle makes electron carrier NADPH, which later brings electron to the electron transport chain. In Krebs makes FADH2, its function is the same. Krebs also makes some ATP, another claim to fame. We're talking Krebs. It's a citric acid cycle, Krebs. Carboxylic acid cycle, Krebs. The cycle makes one ATP, 3 NADPH, 1 FADH2. Right before the cycle's a transitional part, links Krebs to glycolysis, so Krebs can start. Enzymes break a CO2 off a pyruvate, yields an NADPH and acetyl-CoA. Acetyl-CoA carries carbons too, with all the energy that derives from food. It's a highly reduced molecule with energized electrons. It fuels up the Krebs cycle as the cycle's on. Now at the start of Krebs, this acetyl-CoA has the 2 carbons it carries in with the weight. Enzymes put these on oxaloacetate with carbons 4, makes 6 carbon citric acid. Who could ask for more? Notice 3 carboxyl groups on the citric acid. That's why the cycle's also named for tricarboxylic acid, or TCA cycle if it's acronyms that you prefer. Krebs in honor of its discoverer, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. The cycle makes one ATP, 3 NADPH, 1 FADH2. Enzymes work on citric acid and remove the CO2, and other enzymes modify and oxidize it too. The 5-carbon result is alpha-ketoglutarate. Krebs cycle, it's so great. Every oxidation empower the reduction of NAD+, which gains electron carrying function, become an NADH, that energy sensation, which later on in respiration powers ATP creation. Another oxidation follows another CO2 remove, leaving us with a 4-carbon molecule. Another NADH results from this oxidation. Krebs does its energy transformations. It's 4-carbon molecules, it's acetyl-CoA. Still has lots of energy, enzymes can take away. A series of reactions yield 1 ATP, the cell's main energy currency, leaving just enough energy for enzymes to reduce NFADH to FADH2, and 1 less NAD+ will also get reduced as the final electron carrier NADH gets produced. We've harvested what energy came in at Krebs to start. Now we have oxaloacetate at this final part. Oxaloacetate is the commencement and finale, ready to meet acetyl-CoA and here at the final tally. Krebs goes round and round, such an ancient cycle, spinning like the wheels of my bicycle. Krebs like the axle of aerobic respiration. I breathe out CO2 with every exhalation. We walk through the cycle. So now let's review. Input is acetyl-CoA with carbon 2. The carbons can't remove, releasing CO2. Exhaling sends the CO2 out of you. The cycle's functions: energy transformation, 3 NADPH and FADH2 creation, and also synthesis of 1 ATP, which cells directly utilize for energy. For every glucose cells absorbed, the cycle runs 2 times. As long as cells with fuel, Krebs is running just fine. It proceeds electron transport chain and follows glycolysis, expanding round and round in the mitochondrial matrix. Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. The cycle makes 1 ATP, 3 NADPH, 1 FADH2, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. The cycle makes 1 ATP, 3 NADPH, 1 FADH2.
Okay, this is the end. We have to stop here. So we have just talked about respiration, and I'm sure you've appreciated how glucose is used to generate energy. It's quite a long story, so I will have to end it here. But I may have to make some more videos which may explain certain areas, either in photosynthesis or respiration. Okay.